So I'm a chemist by training. I did my undergrad masters in chemistry. So that's where I come from. New to the field of AI, but at least I'm trying to figure out uh, how to use it. Let's call it sensibly in the real life applications, because there are some. So I'll present some facts here where we found that just blind application of AI models in things like, especially in healthcare and drug discovery, it does give a lot of. I mean, there's a lot of things which need improvement. You can't just blindly apply them. So that is something you have to be careful about, and I've focused a lot on these things in the past few years. So I will try to present that. So I'll basically start. Okay, great. Yeah. So impeccable is it's acronym which is not defined here, but basically it came up when we were. So I was basically in my early days of postdoc, and COVID came, and everyone was trying to use computational methods to develop drugs for COVID. So we published this paper in 2021, which is where the impeccable was established. So it is about using mechanistic AI approaches, as I call it now. I didn't use this term at that time, but basically a combination of the traditional physics-based method. When I say traditional, it's not very old. People have been using simulations in drug discovery. There is a field of computer-aided drug discovery (CAD), which is say around 2 decades or around that much old. So there were methods around already. When I started my PhD back in 2014, there were methods there, and then let's say about a decade ago or so, a lot of AI boom came in. AlphaFold was published in 2021, I think, and all these things were happening. So all the pharma companies were sort of running towards using AI models and all those things. So my talk will cover a lot of this. So basically, the way I've structured it is, I'll basically very quickly tell you what drug discovery is all about, right? Because some of you might not have a background in drug discovery. So basically a graph which shows what is drug discovery and what are the challenges there. And then, because we are talking about AI, right, and because I will be talking about a mechanistic AI approach, so I need to first provide you a motivation of listening to me further. So I will tell you what are the applications of AI in drug discovery, and where does it fail, and why does it fail, right? And then I will give you a very quick background of the physics-based methods, because it is required in the impeccable workflow I will be defining and talking about. So I'll give you a brief background about what the physics-based methods, and the most important aspect is I'm not complaining purely about AI. I also complain about the physics-based methods, because there is something called uncertainty quantification, which is an important thing. Because if you are trying to work on a real life application, if a doctor is going to take decisions on clinical decisions about the patient, or if a pharma company is going to shortlist drug molecule based on some computational prediction, they have to know that these are reliable, right? You can't just have some random noise and make decisions based on them. So there is something called uncertainty quantification, which was the focus of my PhD and of my research so far. So I will also talk about why UQ matters, and how I came up with some methods which take care about these things in a more systematic way, and then how, because we were taking care of uncertainty quantification and we had these reliable methods, as I call them, then having including them and combining them with the AI method gave us something which is as good as impeccable. So that's the overall background of my talk. So let's begin first of all. Yeah. So that is it. I hope you can see. Don't worry about numbers, but basically the whole pipeline of drug discovery is shown here. So there's target validation, then you have compound screening, then you have lead optimization until here, and then you have preclinical testing. Until here is all preclinical, and then you move to the clinical trials, which is phase 1, phase 2, phase 3, and then you have launch of a drug. So this whole pipeline, there are estimates which say that it takes about 10 to 15 years for a single drug, and that's the reason we do not have a COVID drug so far, right? COVID came in 2020, already 6 years, right? But we do not have a single drug which is purely for COVID. There was some repurposing, which is that an approved drug was tried to, people tried to use it for COVID. It worked in some cases and so on, but basically there is no pure drug for COVID which is specifically developed for COVID, because the timeline is looking like that, right? That's one thing. The second thing is the cost. There are estimates, and these are old estimates, by the way. I have to update myself, but so unfortunately I could not do it today. But the estimates are that it takes about $2 to $3 billion on an average to just have one drug molecule out in the market. So this is very expensive and a very lengthy process. That is the challenge, right? So now how can we use computational methods to make it better? There were methods, as I said, which were available since last 2 or 3 decades, early 2000 and something. People started using these methods, but again, it has not brought down the cost. It has not made it faster. The same thing happened back in 2015, 16ish, when AI was in the boom, still in the boom, but people have been using AI models into drug discovery with the hope that it will bring down the cost and the time. Again, we have failed, as I'll show. But I am not trying to demoralize anyone. I am just saying that we have to understand why is it not working and make amends to our methods, and then we should hope that it should work. That is the approach I am going to take today, right. So, that is the timeline and things, and what the talk will be focusing today is IMPECCABLE, which is about the preclinical drug discovery. More specifically, the compound screening, the second column, and the third column, lead optimization. That is what IMPECCABLE currently focuses on, and the future is to expand it further, but right now the result file present today is just focusing on these two things. Okay. So, the first part is just to give you a gist, target validation, which is the way drug works is there is in your body, basically when a virus enters, let's say COVID virus enters, there are like 26 different proteins, 29 proteins I think, which it takes into account. So, basically there is a whole process in which the virus replication happens. So, what you do is you choose one target out of those 29 and say this is what I am going to focus on, and then you basically find a molecule which can bind to that protein at a specific location, and such that if it binds, then it will break the whole chain and the virus replication stops. That is how it actually kills the virus. That is the whole funda behind drug discovery. So, you are essentially trying to find a molecule which is binding to a target protein. So, the first step is to identify the target protein, which is the target validation step, which is not IMPECCABLE focuses on. Once you have the target protein, then the next thing is to find a molecule which can do that. So, it is done in 2 stages, basically compound screening and then lead optimization. Again, do not worry about the details, but the thing is that the chemical space is huge, 10 per 60 according to some estimates, that is a large number of molecules which are possible, which are drug like molecules. So, obviously that is a very large molecule space. So, what you do is you perform the initial screening first of all, and then shortlist some molecules, do some testing, and then if you find that they are binding to the desired location on the protein, then you call them hit, and then you try to optimize them, make small tweaks to the structure you found, such that it improves the properties of the drug as you want them to be. That is called the optimization stage. So, this IMPECCABLE focuses on these two, uses some combination of AI and mechanistic approaches to do these things in a more effective and a faster way. That is the idea. Okay, good. A lot of background, and yeah. Now, this before I get into IMPECCABLE, this is just a gist of an overview of how AI is being used in drug discovery. As I said, it has become very prominent in the last 1 decade or so. This is one of the papers. So, basically you can use AI to generate new structures, or say if you have a known library, you can try to develop models which can screen the library and find out potentially good structures. You could model the drug target interactions, see if the molecule binds to the drug or not in the way you want, or affinity, binding affinity is the property, thermodynamic property which we are after, which is used to rank the molecules and say if they are good or bad. So, you can have all those things, and AlphaFold everyone knows, right. So, if you have a sequence in a protein, you have a primary sequence which is the linear chain of amino acids, that is what a protein is, but that is a sequence, it is called a primary structure, but ultimately you want to get a 3-dimensional structure to perform any computation or anything. So, converting a 1-dimensional into a 3-dimensional structure is something which was a challenge. You could perform experiment which may or may not work, but still there is a whole curation of library called protein data bank which people use nowadays. AlphaFold actually used that database, and now it is giving you a capability of generating structures from 1-dimensional to 3-dimensional structures, because you need those to perform any kind of computation. So, just an overview of how AI methods can be used. Now, let us get to AlphaFold, the Nobel Prize winning model and everything, right. People are claiming a lot of things about it. Let me give you some numbers here. AlphaFold 2 has 100 million parameters roughly. It is a very big neural network, obviously. But as is true for any neural network, these parameters do not have a physical meaning, right. They are just weights and biases, and if you want to perform any uncertainty quantification, which is, as I said, emphasize very important, you can't really identify that these are the parameters I want to focus on, because they do not really have a meaning, right. And it is not specific to AlphaFold. Any large language model is basically you are training to find patterns and you are getting some weights and biases, but there is no physical relevance behind those parameters. That's one of the drawbacks, I would say. So then is it really, like, people are claiming that AlphaFold has solved the protein folding problem. I argue against that, because protein folding is a whole process of going from a linear chain of amino acids to the very complicated three-dimensional structures, which is not AlphaFold is able to give you. It will simply, you can input a sequence and it will give you three-dimensional structures. So it is helping you, let's say, overcome or bypass the requirement of experimentally determining three-dimensional structures. It can give you structure, but it can help you with structure prediction. It is not solving the protein folding problem, first of all. Second thing is, whether those structures which you are getting are reliable or not, that is another question, right? Now, because I'm talking about drug discovery. In drug discovery, what you need to have is a structure where we have a small molecule bound to a pocket somewhere in the protein at the binding pocket, as we call it. Now, this is and we know very well, I'll show you some examples in the next few slides, that even if there is a small change in this in the starting pose, let's say if this is a whole big protein and this is the binding pocket, and if the ligand molecule is inside this, if it is like this and you get the binding affinity, it will be something. If it's slightly different, it will be very different value, and it could be as different as that, whether if you're trying to rank the molecule, it could be a very good one or a very bad one, right? You could keep it or lose it. That kind of decisions can be made on just subtle changes. So the point is that even small changes in the starting pose of the protein ligand complex can make big differences in the downstream calculations. So there is a study I've cited here where they found that it can give you, AlphaFold can give you structure, protein structure, that's fine. It could be a reasonable starting point, but if you go down to a very specific binding pocket and see if you can get structures reasonably good, there are studies which show that AlphaFold structures are not very good, right? Because small changes can, and there is no chemical law or physical law behind it. As I said, those parameters are basically just training values, right? I'm sure if you train it again, you might get some different values. You still get the same predictions, but the parameters could be different, right? There are also several other co-folding models. We have tested them all. We have found that there is no agreement. But I'm not going to show that slide here, but we actually found it in a recent study, not published yet, that when you compare the AlphaFold 3 versus Boltz and Kai and Rosetta, and there was another model which we developed, there was RMSD were very large. RMSD is a value which should be small. If it's small, it means there is agreement between structures. There were large RMSDs, even 10 angstroms, in some cases 20 angstroms, which is huge, right? Which means basically all models are giving you some different structure for the same compound, right? So which one do you believe? That's the question. What I'm trying to say is that there's a lot of hype, but the reality is far from there, right? That is the point here. And AlphaFold 3, I don't think we know the actual number of parameters. I don't think they have released the code, but anyway, it will be maybe an order of magnitude larger, right? Now let's get to the point of there's again a lot of hype that there was one company, Accenture. I think I have it in the next slide. Accenture was a company which is overtaken by Recursion now. In 2021, I think it was one of the first companies to claim that we have developed a pure AI drug. I don't know whatever that means. Pure AI drug for COVID, right? And it was under trials, and today we know that it failed the clinical trials. But that is what the claim was at that point. Similarly, there were a lot of other claims. This is one company, I think it's called Benevolent AI. It has now been overtaken or acquired by some other company. But back in 2023, there was a lot of potential it promised, and it was a multi-billion company at some point. But then it failed. And this is just one example. There are a lot of companies which came up. There was a boom, a lot of investment flowing into it. Now I think there's a bubble which is about to burst, and so on. All those things are happening. What I'm saying is the real check is we do not have a single drug as of today which can be attributed completely to AI, not even a single drug, right? I'll show you some snapshots in the next slide, I think. Yeah, this is one snapshot I took. I think some people have seen this before, but so this was taken about a year ago or so. Accenture, no, not Accenture, this is Recursion, I think on the left hand side, this is Recursion, and on the right hand side is in silico, right? Both these companies, they claim to have develop methodologies based on pure AI models and to do drug discovery. This is the status of the various drug candidates they have at the various stages. So there are a few of them in phase 1, phase 2 of clinical trials. By the way, let me tell you, there is a 90% failure rate in clinical trials based on the traditional method, right? So there are things which you can ask, questions you can ask, whether if something is doing good or not. Let's say if AI is adding something or not, whether the failure rate is going down, whether it's moving faster. Do you have a question? Oh, okay. Yeah, whether it's moving faster, whether it's making it cheaper. These are the questions we have to ask, right? Otherwise, how can we say if something is successful or not? But this is the snapshot about a year ago. Not a single drug is, as I said, approved so far. And this is, now you might say that, it's a year-old thing. And if you remember the timelines I was showing in the first slide, it was a 15-year process, right? So what do you think it's happened 1 year down the line? Same thing. Nothing changed. Basically where we were, right? No change in 1 year. So nothing is moving faster. The claims are overhyped. I am, again, let me put a disclaimer here. I am not saying that AI is useless. I am not saying the methods are useless. I am just saying that we have to understand the limitations and work accordingly. Then we will be able to achieve something useful. And I will, I would like to claim that Impeccable is moving in that direction. Why? Because I have some strong evidence which I will show by the end of this talk, that we are getting something usefully out, right? So that is the long background of saying things. Now let's come to, first of all, why should we combine physics-based method and AI-based method for drug discovery? So we know that both types of methods have their own limitations. Physics-based methods, they have no reliance on any training data. Even if you don't have, if it's a standalone complex, you can still, because it's based on physical laws, you just have to solve some calculations which are based on some theory. You can solve them, you can get the properties. For example, binding affinity, I'll show in the next few slides what is binding affinity. But it is one of the properties which is a chemical property, a thermodynamic property, which you want to calculate to perform drug discovery at the initial stages. You have a standalone molecule, doesn't matter whether there is other knowledge or not, you can just get the binding affinity using a physics-based method. So there is no reliance on training data. It is a standalone, it is accurate, right? But the problem is it's very expensive. Performing simulations is really expensive, right? So to get something accurate, and in the way of, if you want to take care of the uncertainties associated with the calculations, then it's even more expensive, because we have to perform ensemble simulations, not a single simulation. So it is expensive. On the other hand, AI methods, they are, once you have a model which is trained and it is, let's say if it's giving you some reliable results, then it's very quick, and the inferences are very quick, right, as compared to the actual simulation. So it's very quick, but the problem is they are relying on the availability of data. And drug discovery, it's a very data sparse process. So there is, first of all, not enough data available. Even if it's there, there might be bias. And we have a lot of evidence from our own research where we show that if it's out of distribution, so if you are working on, say, a type of chemotype, a type of functional groups, or a type of a given type of library of compounds, you move out of it, you're getting something novel, the predictions actually fail, or they become very less reliable. So this is a problem with the AI models. But what the good news is that you can actually combine these two. So what you can do is you can use physics-based method to generate virtual data sets, overcome the problem of the lack of data, remove the bias in data. But the condition is that the physics-based model should be reliable. You should have confidence that whatever they are giving you is something useful, not random noise, not that if you redo the calculations, you get something else. You should have that confidence, and if you have that confidence, then if you generate those virtual data sets and combine these two, it will do something useful. That is what the idea was when we thought about Impeccable about 5 years ago, and I think we are moving in the right direction. Okay, so that was the long background. Now we'll get into the Impeccable, but before that you have to wait a little more, because I'll tell you about the uncertainty quantification on the physics-based method. I'm not going to talk about the physics-based method in detail, just give you what we are after and how do we take care of the uncertainty quantifications. Yes. What time is it? Oh, okay. I'll try to skip them those. Anyway, so molecular dynamics is a method of running simulations for protein ligand complexes. Ligand is a small molecule which is a potential drug candidate or so. So what we know is that if the starting conditions are, okay, I can't. Okay. Anyway, if the starting structures, if the structures are identical, but even the velocities are different, which means the starting point is slightly different. Even in then they diverge. The simulation, the conformation which generate using simulation, they diverge, which means the properties you calculate, in this case binding affinity, they will diverge a lot, which is a known fact. We know it very well, and we care about it a lot. There is a reproducibility crisis in the scientific results. There was a survey done almost 10 years ago where 1500 scientists, they were surveyed, and it was found that 70% could not reproduce anyone else's research, 50% could not reproduce their own research, which is obviously a big problem, right? So we have to take care of it. So we were focusing on how to solve this problem in specifically on drug discovery method, physics-based method based on molecular dynamics. So what we did was, I will very quickly skip through this uncertainty. We introduced ensemble simulations, which is not running a single simulation, but an ensemble of them. The ensemble size is something, it's a tunable parameter. We can adjust it according to the type of precision you are after or the system you are running and so on. But basically you have to run ensemble simulations so that you can get something precise and reproducible, which is why. And what the property which we are after is binding affinity, which is the change in free energy. If those of you from physics or chemistry background, or you should know what free energy is. It's a quantity, thermodynamic quantity, where what we are after is binding affinity is basically the change in free energy. The more negative free energy is for a given process, the more spontaneously a process can occur. So we want delta G values to be negative, and the more negative the better the drug molecule is. So what we calculate is delta G value for large number of drug molecules, library of drug molecules or potential drug molecules, and to a given target protein, and rank them based on their binding affinities. The more negative the better. So we shortlist them and so on. So this is what we do, and just skip those things. Basically, as max and ties, just remember these two words, were the good methods, like say reliable methods, which we wanted to start with. Now we come to the impeccable workflow. It is, I'll just go to the next slide because that contains more details. Yeah. So basically what we found was, in the whole drug discovery preclinical process, we found 3 areas where we could use these machine learning models to make things better. One of them was to have the initial screens. We have large libraries of compounds, billions of compounds. What we want is to screen them down to manageable number, say a few thousand, so that we can use more sophisticated methods. So what we use is something called docking. Docking is not very accurate method, but at least it's good to find out whether a molecule fits into target protein or not. That's the only thing. So we want to do it faster. It's relatively cheap as compared to simulations, but still it's not very cheap when you're running at billions of at a scale of billion molecules, right? So what we want is a machine learning based surrogate model which can predict docking scores very quickly. So this is one of the applications which we identified in Impeccable. In the next slide, I'll show the whole pipeline, but right now this is just the overview of what are the 3 different models we have developed. And the second thing is pose optimizer, machine learning based pose optimizer model. This is again a very big problem. There can be a whole different lecture on this specific problem. We have a paper on this this particular model, pose optimizer. But as I was saying earlier, even a small change in starting pose is going to make huge differences in the binding affinity. So we want to start with something optimal, but we don't know what it is, right? So we need something more efficient there. That was the second thing which we targeted, and the third thing, which is the most important, and I call it the heart of the Impeccable as of now, is the generative AI method, where it can generate new structures rather than just shortlisting a set of molecules from known libraries. We want to generate structures more efficiently, which have properties we are after. And the most important property in this case is the binding affinity. So we want to generate structures which are, let's say, not known in the chemical libraries of, let's say, 2 billion or 100 billion, whatever we have right now, and but they still have they can optimize. So we move in the if this is a big chemical space, we want to move in the directions or in the areas of chemical space, regions of chemical space which have such functional groups or such structures which can lower the binding affinity. That is the idea. So this is very important. So now I'll in the next few, so let me give a couple of minutes of this. So this is the actual implementation. This is something it's under review right now. But it's available online on archive. So we start with, if you can see, can you see the cursor now? Okay, good, right. So basically we start with library, chemical library. Right now the largest one is 100 billion compounds. This work was done on zinc 20, which is 1 billion of a subset of that library, which is reasonable molecules, let's say. So we start with that. The first thing is, if you remember, we want a docking surrogate model. So we randomly sample 1 million compounds, 0.1% of the whole library. So we are 3 orders of magnitude less compute. We perform docking on those and then train this model. The R square values are over 0.9, which is reasonably good, right? And then we use this trained model to screen the whole library. So we have reduced the compute a lot, but we've still gotten a very good accuracy and got a short list of compounds. Then we start with top 10,000 compounds from this short list, and then there is a two-way cycle, iterative cycle going on here. So on the one hand, so whatever molecules we get, on one side we are optimizing the poses. So we start with 10,000 and then we generate, to train this model on the left hand side, this one, the pose optimizer model, we generate 100 poses for each molecule, 10,000 multiplied by 100, 1 million data point, that is a starting training set for us. We train the model and then it is able to predict better poses in the next round of iterations. On the right hand side, the model is about to generate new structures which have binding affinity. So we start with 10,000 molecules. It is a model by AstraZeneca. So we use that as a prior model. It is able to generate structures. What we want is it to generate structures. We want it to learn to generate structures which are relevant for this target protein, which have low binding affinity for this target protein. So we start with the training set of 10,000 compounds and corresponding Asmax simulation. So we perform proper Asmax simulation, very large compute, but we were able to get reliable binding affinities. So there is a structure, there is a corresponding binding affinity, and we use that as a training set, perform reinforcement learning on this reinvent model, and then so that it's able to generate structures which are better binders. And then this loop goes on in both directions. So this goes on, it's an iterative cycle, and by the end of it, we have shown in this paper which is published already. There's a paper published in 2024 and the second one is follow up now. So it's an ongoing process basically, but we have shown that it is first able to generate good binders. So every iteration, when we plot the distribution of binding affinities, we get more and more negative values, more and more negative binding affinities. And the second thing is novelty. So I think I should show that in the, I can skip that one. Yeah, let's see the important. Yeah, okay, this is what I wanted to say. This is one for the 3CL Pro COVID target. What you see in the red distribution is the original seed compound, 10,000 compounds and the corresponding binding affinities. They were, the distribution of those, and then every iteration we used a batch size, two different batch sizes, 500, 250. Every iteration you see the distribution of the binding affinities for the next cycle, and you see it's moving towards more negative values, right? So that was the aim number one. You want more negative binding affinities. We were able to achieve that. This is for one system. We have similar plot for the other system. It's in the published paper. The second thing is we want novel compound, right? So here you see the TSNE plots of Morgan fingerprints. Simply speaking, it is basically showing what is the structural diversity of the compound we had. So the blue cluster, the big blue cluster is the 10,000 compound we had originally, and then every iteration you have a different colored clusters. You can see it's moving away from the blue cluster. So the point is that we are getting genuinely novel structures. That was the second thing. So now with this approach, we were able to show that we have achieved both novelty and optimization. We want to include multi-optimization problem. So this was just a single optimization binding affinity. What we want is, because as I was saying earlier, the clinical trial failure rate is about 90% or so. Binding affinity, good binder thing is 1 point, but you have a lot of other properties you want in a drug molecule. So what we want is to reduce that failure rate at clinical trial stages. We want to perform a multi-optimization in this iterative workflow such that it has all the properties we are after, so that by the end of this iterative cycle, the structures which we are able to get, they have all the properties which a drug molecule should have, so that the clinical trial failure rate will reduce. So examples are, they should be less toxic. They should not have a lot of off target. So we want to be selectively binding to a given target and not binding to anything else, not other proteins, right? Otherwise it will have side effects, right? It should be able to penetrate your cell wall. It should be able to excrete or metabolize in your body, and all those properties are required. So there are ways of quantifying those things, and now what we are moving towards is having this impeccable multi-optimization objective optimization problem, where we add all those properties, try to optimize them at the same time, and hope that it will improve the clinical trial failure rate, and so on. That is the idea. And if I have another couple of, and this is the next stage of impeccable, 2 more minutes, okay. So I think I'll just finish it off. So basically that was the original, if you remember, there was a stage about compound screening. So that is what we are focusing so far. The next thing is lead optimization. That is a similar approach, but not exactly same, because what we are after there, we have a molecule which is a known binder, but we want to make small structural variations in that molecule and see if those structural variations are changing the binding affinity in the right way or not, making it more negative or not. Which means we are not focused on delta G values now, we are focused on delta G values or the relative binding affinities. So for that, equivalent to Asmax, we have something called ties, which is again a very accurate method based on ensemble simulations. We have confidence in the method. So, we use this and perform the same iterative cycle, reinforcement learning, trying to generate new structures in moving in the right direction. And using this approach, we had this time we did not stop at just computational predictions. The paper which is under review right now, it has experimental results. So, we collaborated with some experimentalists. They were able to, so we generated some about 100 structures or so. Then they shortlisted about 6 of them. So, we took the top 10 of based on our predictions. They chose 6 of them, synthesized them in laboratory, and performed the binding assaying, got the experimental binding affinity values, and we found that in the jargon of binding affinities, we were at micromolar binding affinity. We want to go down, we went to nanomolar, order of magnitude improvement, which is very significant in just one shot. So, this is why we are quite excited about IMPECCABLE, and not just us, a lot of our collaborators are excited. So, we are building upon that. But what I am trying to say is this approach worked quite well, because in just 6 out of 1 was already a nanomolar. We came down from micromolar to nanomolar, which is a big deal in drug discovery. So, this is the result which we are hoping to get published very soon, but it is very exciting, and we are already building up on new collaborations and trying to apply this IMPECCABLE. So, for future, if any students are interested, IMPECCABLE is one thing, and the second thing is the mechanistic AI approach. We want to develop that and not have a molecular level, just a molecular level model, but a multi-scale model for disease modeling. So, cancer is something I am trying to focus on, develop a cancer patient digital twin using same mechanistic AI approach. So, that is another area of research which I am trying to focus on. And with that, I will stop and I will take any questions if you have. Thank you.